Uh, 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 uh.
Hello, sir. It has already started.
It has already started, sir. Anoop,
Yes,
sir.
it is not visible here because
Are you
our
sure,
session,
sir?
it actually delays a lot. It is giving a delay of around 20 seconds.
Okay, okay. So, hello guys, how are you all?
We both are here.
Anoop sir is going to teach us spermatogenesis today.
Well, I'll be able to give you certain chemicals which are there present on the sperms, and that is how they work.
Right.
Because it is all, you need to understand that
The chemistry.
biology at the
Bilkul.
level is organic chemistry only.
Undoubtedly, sir, undoubtedly. Chemistry is the basis of the life. I always tell you that biology is nothing without chemistry. Right, guys? Agree?
So today, Sachin sir is starting the class a little early, and I would be starting the class a little late because in our studio there is a power cut. So my class would be starting at 5 p.m. and Sachin sir has already started the class.
Great.
Sir,
So all of you can see me, you can hear me, audio, video, everything is clear. Please let me know in the comment section, in the chat box. All of you can see us clearly, you can hear us clearly. Anoop sir, what's the topic which you are going to take today?
today I'm going to take the very important topic in concentration unit, that is solution chapter. The concentration unit, we are going to talk about molarity and molality.
Okay, I always used to get confused between this molarity and molality. I hope Anoop sir will clear all your confusions. So Anoop sir, at what time you will be taking your
It's
session
going to be
today?
today at 5 p.m.
Your session
Otherwise,
is
we
at
have
5
the
p.m.
session at 4 p.m.
In
but
routine,
today
you
because-
take at 4 o'clock. Na? So today, Anoop sir will be taking his session at 5 p.m. So guys, please, all of you have to attend the session live. So thank you so much, Anoop sir. Right?
You are most welcome, and thanks for allowing me to be here.
My pleasure, sir. It's always a
All of us
pleasure to share the screen with you.
Like.
Hit the like button.
Right.
Okay, guys. So wish you a very, very bright and prosperous future. So I'm your very own Dr. Sachin Kapur, right in front of you. And today I'm going to discuss a very important topic from human reproduction, that is spermatogenesis. We will understand how the sperms are produced. So keep hitting the like button. All my Raja beta and Rani betiyan, याद रखिएगा वर्ल्ड फेमस डॉक्टर बनना है. So world famous doctor banna hai, then you have to work hard. Always I carry one quotation in my class so that I can motivate you, and what I have today is, remember, a diamond is just a piece of coal that did well under pressure. How beautiful it is. Coal and diamond both are made up of what? Carbon only. So diamond is just a piece of gold, but it did well under pressure. So if you are able to tolerate this pressure, if you are able to go through these adverse circumstances, which you might be feeling right now, that you are preparing for such a prestigious examination and there is a lot of pressure on you people, so please understand that you will be converted into a shining diamond. All my Raja beta and Rani betiyam. Hello Sonu, hello Divya, hello Dev, hello Rohit, how are you? Masroor Ahmed, God bless you all. Wish you all a very bright and prosperous future. So let's proceed forward. I'm your very own Dr. Sachin Kapur with MPhil and PhD degree with more than 24 years of teaching experience, and fortunately I got this what should I say, opportunity to teach students like you, to mentor teachers like you. So more than 10 lakh students and teachers have been mentored by me. So it was a kind of joint effort, their hard work and my contribution, and today they are at a very successful position in their life, and the same I expect for you people also. Mohammad Rehan, Prerana, Sandesh, Ananya, God bless you all. Wish you all a very, very bright and prosperous future. So all my Raja beta and Rani betiyan, today what I am going to discuss is about sperm production in the testes. In our previous session we have discussed that where are testes located and why testes are located in scrotum, because sperm production, maturation, storage and survival requires 2 to 3 degrees lower temperature than the core body temperature. That's the reason that testes are located in the scrotum. Today we'll be discussing that how these sperms are produced, and once this process of spermatogenesis begins during puberty, it continues throughout life. You know that a male continues producing sperms throughout his life. though with advancing age, the number of healthy sperms produced reduces. So, Unit 6. Class 12th NCERT, the name of the unit is Reproduction, Human Reproduction, right? So, what are the different chapters in this unit? Reproduction in organisms. Chapter 2 is sexual reproduction in flowering plants. Chapter 3 is the one which we are discussing, that human reproduction. And after completing this chapter, I will be discussing what? Then I'll be discussing reproductive health. Okay, that's also a very interesting chapter. So this is how it looks like when you open up your NCERT books. This is what you will find. So just to give you a feel of the NCERT. Okay, let's have a quick recap. Are you all ready? Tirthraj, Radhika, Akshit, Debesh, Sonu, Priya. Are you all ready for the session? Can I start? Let's have a quick recap of what we have discussed in the previous session. See, male reproductive system, it is located in the pelvic region. So this is a side sectional view of the male reproductive system, which is located in pelvic region. In my previous class, I told you people that in male reproductive system, we study about the primary sex organs and the secondary sex organs. Primary sex organs are also called gonads. Why they are called primary? Because they are directly involved in the process of sperm production, gametogenesis. Testes produce sperms and they also produce the male sex hormones, which are collectively called what? Androgens. Masoor Ahmed beta, I have covered this topic already, that testes are covered by tunica vaginalis, tunica vasculosa, tunica albuginea. The coverings of the testes I have already discussed in my previous lecture. Secondary sex organs, these are not directly involved in sperm production. What is their function? Their function is storage of the gametes, maturation of the gametes, transportation of the gametes. We'll be discussing all the secondary sex organs in detail. Now, we discussed this point that why testes are located in scrotum. Testes are located in scrotum because sperm production, maturation, storage and survival of the sperms requires 2 to 3 degrees lower temperature, right? It is not only sperm production, Devansh, it is about the storage of the sperms that also requires low temperature. Maturation of the sperms also requires lower temperature. So say this is scrotum, just a rough representation. This is scrotum. There is a partition called scrotal septum. This region is what is called as inguinal canal. This region is what is, shall I write here? It is inguinal canal. Testes communicate with abdomen through inguinal canal, right? There is a slight difference in the position of the right and the left testes. Left testes hangs slightly lower than right, so that when both the legs are very close together, testes should not get crushed. But there is no hard and fast rule. Both testes can be at the same position, or even the right could be hanging down. It doesn't affect the sperm production in any way. Is it clear? Ram Gopal, Devansh, Sonu, Vashu Pundir, Priya, Namrata, hit the like button and please share the session with all your friends. I always say, Vidya bantane se badhti hai. Okay, chaliye ji. Let's see this diagram also. If you remember, I told you about spermatic cord. Testes, they hang in the scrotum with the help of spermatic cord. Spermatic cord has blood vessels, lymph vessels and nerves. Blood vessels, lymph vessels and nerves, right? It also has vas deferens, which is called ductus deferens. What is the function of ductus deferens? Testes are producing the sperms and vas deferens carries those sperms to outside. We'll discuss vas deferens in detail. Inside the testes, you can see these chambers which are called lobules, right? Someone was asking me about the covering of the testes. So I told you that it is tunica albuginea which covers the testes. At certain places, tunica albuginea enters into the testes and forms the compartments. What are those compartments called? They are called testicular lobules. Can anyone tell me how many testicular lobules are present in one testis? Very simple question. How many testicular lobules are present in one testes? Sonu, Ananya, Sumit, Arpita, Namrita. Very easy question I have asked you. Suppose this is testes, है ना? And this is what is vas deferens. Tunica albuginea enters into the testes and forms compartments or the chambers. What are these chambers called? Testicular lobules. Gyandeep has said right, 200 to 250. बिल्कुल सही, एकदम सही कहा है आपने. राधिका, देवांश, प्रेरणा, नित्या, नम्रता, राम गोपाल, राधिका, clear. Mohammad Rehan, 200 to 250 testicular lobules. And inside the testicular lobules, you find what? Seminiferous tubules. Seminiferous tubules, highly convoluted, highly folded. Length of one seminiferous tubule is roughly 70 centimeters. 70 centimeters. Now see here, each testis has 200 to 250 compartments. What are the compartments called? Testicular lobules. Remember, this can be framed as a question. Each lobule has how many seminiferous tubules? 1, 2, 3. 1, 2, 3 seminiferous tubules. I hope you are liking the diagrams. Seminiferous tubules jo hain, these are structural and functional units of testes. Clear hai? Very good. Let's proceed. Another beautiful diagram. This structure is testes. You can see a cut view, and inside the testes you can see what? You can see seminiferous tubules. Highly convoluted means highly folded. They open into straight tubules which are called tubuli recti. Tubuli recti open into retitestes. Retitestes, this structure, this network which you can see here, this is retitestes. Fine ciliated tubules originate from retitestes, and those ciliated tubules are called vasa efferentia. Shall I write here? What are they called? They are called vasa efferentia or efferent ductules, right? So efferent ductules carry the sperms towards what? You can see there's another highly folded tubule, and that is called epididymis. You should be able to pronounce the terms properly and write the terms, write the correct spelling. So epididymis, the head region is caput epididymis, the body is corpus epididymis, and the lower region is the cauda epididymis. It's okay if you don't remember the different names, still remember this much, that epididymis, this is going to store the sperms. Is this diagram good? Are you liking the diagram? Let me know in the comment box. Did you like the diagram? Is it opening the gyri and sulci of your cerebrum? As I say in Hindi, dimag ke kapat khul rahe hain. Come on guys, let me know, and keep sharing the video lecture in all your groups, all your academic groups. Sumit, Sameer, Prerana, Tirathraj, Deepu. Great. Prerana says that length is 70 centimeters. That correct. 70 centimeter jo hai lambai hai seminiferous tubule ka. In my previous session also I told you, inside seminiferous tubules, when you look inside the seminiferous tubule, you find that there are two types of cells. And what are those two types of cells? Germinal cells, which are going to form the sperms. What are they called? Spermatogonia. What are they called? Spermatogonia. Second is somatic cells. And what are they? Sertoli cells. There are different names for sertoli cells. They are also called sustentacular cells. What are they called? Sustentacular cells, supporting cells, nourishing cells, because they provide structural support, protection and nutrition to the developing sperms. So they are sustaining the developing sperms, and that's why we call them as sustentacular cells. Clear hai? Sachin, you have been repeatedly telling me that I should change the background, and I always forget to do that. Okay, I'll ensure in the next class. You guys can get connected with me on my Telegram channel also, that is Dr. Dr. Sachin Kapur, Kapur Talk, Talk, Dr. Sachin Kapur Talk. And please do not forget to see the links which are mentioned in the description are NEET Elixir Telegram group also you have to join. Okay, Ananya Sarkar, I promise you that from next session onwards I'll be carrying the slides with a white background, if that is okay to you people, right? Okay, hit the like button to mark your attendance in my heart as well as on my channel. So all my Raja Beta and Vani Betiyam, I told you two types of cells are present inside the seminiferous tubule, sertoli cells and spermatogonia. Spermatogonia will form sperms. Now, this is another beautiful diagram. You will get a complete PDF of the session. Don't worry. I'll provide you the PDF of this session on the Telegram group. So, all these diagrams I'll be presenting to you people. Right, Ji? Clear hai? Chaliye. Let's proceed forward. Another beautiful diagram, cut section of the testes, and here you can see one seminiferous tubule in an enlarged way. So these grey colored cells are what are? Spermatogonium. When I say spermatogonium, it means one spermatogonia, singular spermatogonium, plural spermatogonia. And you can see sertoli cells also. The boundary of the sertoli cell is something like this because the developing sperm are embedded in the membrane of the sertoli cell. So is it clear? The transverse section of a seminiferous tubule. Then here you can see a small portion of the seminiferous tubule is enlarged. Small portion of the seminiferous tubule is enlarged. You can see the different stages of the development of the sperm. You can see the spermatogonia. Then you can see primary spermatocytes, secondary spermatocytes, spermatids, and then finally spermatozoa. Right? What are the different stages which are able to see here? Spermatogonia, primary spermatocyte, secondary spermatocyte, spermatids and sperms. This is what we have to discuss in detail today. Right? Clear hai ji? Okay guys, chalo, let's proceed forward. Develop an interest for your subject. Love your subject. See the beautiful diagrams I carry for you people. Such an enlarged view which makes everything crystal clear. The diagrams given in NCERT are not of very good quality. The print is also not very good, right? So that is what my aim is, to make the things more presentable to you people. So see, my Raja Beta, this is enlarged view of a small portion of seminiferous tubule. This is the lumen of the seminiferous tubule. Can you see spermatids which are embedded? They are having these tail-like structures also. They are getting converted into sperms. So they are embedded into the sertoli cells. In Hindi you can understand, sertoli cell mein dhase hue hain. And when they will be released from the membrane of the sertoli cell into the lumen of the tubule, that process is called... Can anyone tell me what is that process called when these sperm are released from the membrane of the sertoli cell into the lumen of the seminiferous tubule? This question has been asked in NEET 2022 exam. Here you can see a transverse section. Here you can see an enlarged view. You should hit the like button. You should hit the like button, my Raja Beta and Rani Betiyam, because it requires so much of hard work to make these slides for you people. Anji, bataiye. Sachin says, what is the function of Leydig cells? Sachin, Leydig cells are also called interstitial cells. They are the endocrine cells. These secrete the male sex hormones which are called androgens. Spermiation. Sumit has given the right answer. The process of release of sperm from the membrane of sertoli cell into the lumen of seminiferous tubule, that is spermiation. Spermiation. Question can be framed from this. Right. Tirath, Sameer, Prerana, Arpita, Gyanadeep, Radhika, Divya Khushwaha. God bless you all. Okay, ji. Let's proceed forward. This diagram is from NCERT. Now you can say, see, the diagrams which I have shown you just now and the diagram of the transverse section of the testes or the tubule which is mentioned in NCERT, Things are not very much clear. So my aim is always to make the things clear to you people. Raja beta, this diagram has been taken from NCERT, but of course we cannot ignore it. Examiner can give you this diagram and they can expect you to know the labeling. Which is sertoli cell, which is spermatogonia, which are the sperms, though everything appears to be blur and hazy. That is how the diagram has been presented in NCERT, right? But it is important. Labeling is important. Okay, guys, what are the interstitial cells? Interstitial cells, which are also called Leydig cells. Someone was asking me, what is the function of the Leydig cells? So, Raja beta, interstitial cells, also called Leydig cells, these are located outside the seminiferous tubule. Suppose this is transverse section of testes, one seminiferous tubule, another seminiferous tubule, another seminiferous tubule. So outside the seminiferous tubule, these spaces are called interstitial spaces. These spaces are called interstitial spaces. Here you have blood vessels also. Now in addition to blood vessels, you have endocrine cells which are called Leydig cells. So one question is, location of the Leydig cells. Are they inside the seminiferous tubule? No. They are not inside the seminiferous tubule. Leydig cells or the interstitial cells or the endocrine cells of the testes are outside the seminiferous tubule in the interstitial spaces. And what is the function of these interstitial cells or Leydig cells? It's written on the board. What is the function? Sumit, Ananya, what is the function? Ananya Sarkar beta, the function is synthesis and secretion of the male sex hormones. These produce male sex hormones which are collectively called androgens. Male sex hormones are collectively called androgens. Can you give me an example of androgen? Any one of you? Give me an example of male sex hormone. Sumit says, right, testosterone. Androgen is a category of steroids. These are steroid hormones, and an example of an androgen is testosterone. So these are steroid hormones, and I have told you earlier also, cholesterol is the precursor for steroid hormones. Your body has so many steroid hormones. Females also have steroids, estrogen, estrone, estriol, estradiol. So they also they are formed from cholesterol. Cholesterol is the precursor, right? Okay. So testosterone is an example of androgen. There is dihydrotestosterone, androstenedione. There's so many types of androgens. Okay, ji. So is it clear about Leydig cells, interstitial cells, right? You know, we have pituitary gland. Pituitary gland produces gonadotropins, LH, FSH. LH is luteinizing hormone. FSH is follicle stimulating hormone. Pituitary gland releases these hormones into your blood circulation. Luteinizing hormone LH is also called ICSH. If you remember the full form of ICSH, interstitial cell stimulating hormone. So which cells of testes, they have receptors for ICSH? Interstitial cells, they have receptors for ICSH, which is secreted by anterior pituitary. So pituitary will release the gonadotropin into your blood through blood circulation. This ICSH, which is also called LH, it will reach the testes, and inside the testes it will act on interstitial cells. So it is in response to this LH or ICSH that Leydig cells, they synthesize and secrete androgens. Is this point clear, Divya? This is how the endocrine functioning of the testes is regulated. You are understanding the things? Enjoying the session? Okay, can you proceed forward, Divya? Okay, next slide. We are discussing structure of testes, and next I am going to tell you about spermatogonia. Singular is spermatogonium. I told you, spermatogonia are the germinal cells. They are very sensitive to temperature. If the temperature is high, spermatogonia gets destroyed. That is the reason the testes are located in scrotum for a favorable temperature. Stem cells called spermatogonia, singular is spermatogonium. They develop from primordial germ cells that arise from the yolk sac. Now this must be something which is technical for you people. I will explain about yolk sac also when I will discuss embryology. I hope you have heard of the extraembryonic membranes, amnion, chorion, yolk sac, allantois. तो germ cells जो होते हैं ना, our body has two types of cells, somatic cells and germinal cells. So these germinal cells, the primordial germ cells, they arise from the yolk sac. That means they are not originating in the testes. Their origin is extragonadal. They have migrated from yolk sac into the testes. Is it clear, ji? Primordial germ cells arise from the yolk sac. Now, these enter into the testes during 5th week of development, when the testes of the male baby are developing, when the male baby is inside the uterus of his mother. During the 5th week of development, from the yolk sac, the primordial germ cells have entered into the testes. So in the embryonic testes, means when the baby is inside the uterus of his mother, primordial germ cells differentiate into spermatogonia. These spermatogonia remain dormant during childhood. They are inactive. They are not forming the sperms. In males, sperm production begins during puberty, when the childhood period is over. Sperm production starts. Males attain puberty around 12 to 14 years of age, and then the sperm production starts. So it's written, actively begin producing sperms at puberty. Right, Ji? So there are PGCs, primordial germ cells, which are forming spermatogonia. And primordial germ cells do not develop inside the testes. They develop in the yolk sac of the embryo. Extragonadal origin. They migrate to testes during 5th week of the development. Right? Ananya says, sir, आपने इस chapter को super easy बना दिया. Ananya, that is what I want. I want to give you thorough information, genuine information. I want that you should develop interest for the subject. Do not limit yourself to those pages of NCERT only. You have chosen medical sciences as your career. It requires so much of in-depth study. So have a strong foundation. Develop love with English language, because in English it is easy to convey the information, right? The sooner it is, the better it is. Priya says, what is yolk sac? Priya beta, yolk sac is an extra embryonic membrane that we will discuss in embryology. I will tell you about different extra embryonic membranes. Let's proceed further. Spermatogonia. Toward the lumen of the seminiferous tubule are layers of progressively more mature cells. Now what does it mean? Suppose this is one seminiferous tubule, single seminiferous tubule in a sectional view. This is one sertoli cell. This is another neighboring sertoli cell. This is another sertoli cell. Okay? Sertoli cell, the plasma membrane is highly folded. So these are sertoli cells. Now I'll tell you where are spermatogonia located. Let me use a different color for them. Okay, which color should I choose? Okay, red color. One spermatogonium, another. Okay, this is the nucleus. So between the adjacent sertoli cells, spermatogonia are located. So the center is the lumen of the tubule. Spermatogonia will change into primary spermatocyte, secondary spermatocyte, spermatid, and they will keep moving towards the center or the lumen of the tubule. And finally, you will find that sperms, they are present. Just a minute. Finally, you will find that the sperms, they are present, and their head is embedded in the sertoli cell membrane, and when they will be released from the membrane of the sertoli cell, that process is what is called as spermiation, right? Sumit कह रहे हैं, sir, you are my guru and I am your shishya. Pranam Guruji. Pranam ji, namaskar, God bless you. आप अच्छे से पढ़ाई करें और वर्ल्ड फेमस डॉक्टर बने. Okay guys, towards the lumen of seminiferous tubule, there are layers of progressively more mature cells. So spermatogonia mature hote jate hain and they keep moving forward. In order of advancing maturity, remember, this sequence you have to remember, there is, what's wrong? Why I'm not able to write? What has happened? Let's select the pen again. Okay. Primary spermatocytes which change into secondary spermatocytes which change into spermatids, and then finally sperm cells. So this sequence you have to remember. All my Raja Beta and Rani Betiya, this sequence you have to remember. What is the sequence? Shall I write here? First, there are spermatogonia, right? Spermatogonia, they change into primary. Keep noting in your notes copies also. They keep changing into primary spermatocytes. From primary spermatocytes, which is the next stage, secondary spermatocyte. Secondary spermatocyte change into spermatid, and spermatids change into sperms. Dipanvita, is it clear? Spermatogonia towards the periphery of the tubule. And then as they are developing, they are moving towards the lumen of the tubule, and then they will be released inside the seminiferous tubule. That is what is permeation. Now, sequence you have to remember, right? Sometimes they ask you a question, which is the proper sequence of the development of sperm. Spermatogonia, primary spermatocyte, secondary spermatocyte, spermatids and sperms. Yaad rahega sequence. Dipanvita, Ananya, Mohammad Rehan, Gurdeep Thakur. Yaad rahega. Hit the like button also. Those of you who have not pressed the like button. So much of energy is consumed when we people teach you in this studio. There's so much of heat which is generated by this board. It's a huge smart board. It generates heat also. Though the studio is air conditioned, but still there's so much of heat. So if the teachers are taking so many pains for you people, you should also reciprocate. You can show your love and respect and your affection by hitting the like button, by sharing the videos, and do not forget to comment also once the video is uploaded on the YouTube. In the comment section, I always read your comments and I reply also. I hope you'll agree to that. Arima says primary spermatocytes are haploid or diploid. I'll discuss that. Spermatogonia diploid. Primary spermatocytes are also diploid. Then meiosis 1 happens. Secondary spermatocytes, they are haploid. Right? So diploid, diploid and then haploid. I'll explain that. Don't worry. This is very important. This question has been asked in NEET 2022. After a sperm cell has formed, it is released into the lumen of the seminiferous tubule, and that process is spermiation, right? Plural terms for the sperms, we say spermatozoa, sperm, they are plural. And single sperm ke liye we write spermatozoon, one sperm. But when you have simply written sperm only, so that is plural because sperms are produced in millions. One sperm is spermatozoon, right? Ananya is such a good student. She says, sir, pani pee lijiye. Thank you. Okay guys, spermiation yaad rahega. This is very important. What is spermiation? Process of release of the sperm from the membrane of sertoli cell into the lumen of tubule. Clear hai ji? Spermiation. I want to, Devansh, Malik, Namrata, just write the word spermiation in the chat box right now, so that I get this assurance that you people have understood what is spermiation. Spelling bhi galat nahi karna. Okay ji, let's talk about these cells, sertoli cells, important, right? They are also called what? Sustentacular cells, supporting cells, right? If I show you one sertoli cell, so the plasma membrane is quite folded like this. So this is one sertoli cell. This is spermatogonia, primary spermatocyte, secondary spermatocyte, spermatid, and this is one sperm which is embedded into the membrane of the sertoli cell. So the plasma membrane of sertoli cell is highly convoluted. One is this point. They extend from the basement membrane to the lumen of the tubule. So this side is what is the basement membrane, and this side is what is lumen of the tubule. So they are extending from the basement membrane towards the lumen of the tubule, and there is another name for these cells, and that name is what? Nourishing cells. Why do we call them nourishing cells? They are providing nourishment. Sertoli cells, they are very clever. They know that spermatogonia cannot tolerate high temperature. So blood never comes in direct contact with the germ cells. Germ cells say, blood, please don't touch us, right? So sertoli cell says, okay, don't worry. I will take nutrition from blood, oxygen from blood, and I will pass on that oxygen and nutrition to the developing germ cells. So they are forming a kind of a barrier between the developing germ cells and the blood. That is called blood testis barrier. Please remember, sertoli cells are forming blood testis barrier. Mohammad Rehan, Arima, Ananya, Sumit Roy. Great G. Keep hitting the like button also. Okay, my Raja Beta and Rani Betiya. Here what you can see, spermatogonia which are diploid, 2n number of chromosomes. Then they undergo growth phase to form primary spermatocyte, which is also diploid. Primary spermatocyte undergoes meiosis 1 to form secondary spermatocyte, so it is haploid. Someone was asking me about the ploidy level, so this is clear here. Then secondary spermatocyte, they undergo meiosis 2 to form spermatid. Spermatids are changed into the sperms. That process we'll discuss in detail, how they are changed into the sperms. See, what are the functions of sertoli cells? Sertoli cells, they perform so many functions. They provide structural support, nutrition, and protection to the developing sperms. They are responsible for fulfilling all the requirements of the developing sperm. So it is the duty of the sertoli cells. Second, they are forming blood testis barrier. In short, we call it BTB. What is blood testis barrier? I told you, blood has never seen germ cells. So if blood touches the germ cells, it will destroy the germ cells by immune reaction. So sertoli cell says, okay, blood, please don't touch our germ cells. We are going to form a barrier between blood and the germ cells. So blood testis barrier is something very important which is formed by the sertoli cells. Sertoli cells secrete क्या करते हैं? They secrete ABP, androgen binding protein. They secrete inhibin. This is mentioned in NCERT. Important. Inhibin has negative feedback effect with FSH of anterior pituitary. Pituitary produces FSH. Testes produce inhibin. Inhibin through blood circulation acts back on the pituitary and tells the pituitary to decrease the production of FSH. So this is a kind of feedback loop which is operating. They also secrete MIS, Mullerian inhibiting substance, which is also called Mullerian inhibiting hormone, which is also called Mullerian inhibiting factor. This is also secreted by the sertoli cells. And what is the function of Mullerian inhibiting factor? This I will discuss in embryology also. See, when a fetus is developing in the uterus of the mother, it is in a kind of bipotential stage. It can develop into male as well as into female, irrespective of XX or XY chromosomes. So it is the presence of this MIS which causes regression of the Mullerian ducts. Now what are Mullerian ducts and Wolffian ducts? That I'll discuss in embryology. So simply remember, Mullerian inhibiting substance, it causes regression of Mullerian ducts in male embryo, whereas in female embryo, the Mullerian ducts develop into the fallopian tubes, parts of the female reproductive system. Males के case में सुमित बेटा, MIS causes regression of the Mullerian ducts. Is it clear till now? Whatever I have taught, is it clear to all of you? Any doubt, any question you want to ask, please feel free to ask. Come on, quickly. Jaldi se. Vashu Pundir, any question you have in mind? And please subscribe the channel also and hit the bell icon. Tell your friends also to subscribe to this wonderful channel where quality education is free of cost by experienced teachers. Crystal clear? अच्छी बात है. अनन्या कह रही है clear है. अच्छा, the kind of English which I am using during these sessions, or the way I am speaking, is it clear to you now? It's not very tough English, very simple English. When you will be studying in your medical college, you will be communicating with your colleagues, with your friends in English only, with your professors also in English. Your medical college professors will also teach you in English. So please develop confidence for this language. Right, Akshita? Devansh, I said ABP is androgen binding protein. It binds with androgens. It binds with androgens. Androgens are the male sex hormones. So it binds with testosterone and maintains high concentration of testosterone inside the tubule, and that high concentration is required for normal sperm production. Everything is written here. Everything. Okay, I have simplified the stuff. ABP function, it binds with androgens and maintain their high concentration inside seminiferous tubule. High concentration of androgens is required for normal sperm production inside the tubule. Okay? Inhibin ka kaam kya hai? Inhibin has negative feedback effect with FSH of anterior pituitary. One question you have solved, 4 marks for you. Mullerian inhibiting substance, what does it do? It causes regression of Mullerian ducts in the male embryo. You will get all these PDF sessions, don't worry. Where you will get? Follow me at my social media, Dr. Sachin Kapur Talk, or even Unacademy NEET Elixir channel. All these PDFs will be provided to you. Prerana says, your voice and your English is so good that anyone can understand, sir. We love your voice. Thank you so much, Prerana. मेरी आवाज ही पहचान है मेरी. आगे बढ़ते हैं. Next question. Next, what do we have? Okay. So blood test is barrier के लिए this enlarged diagram. This is for you. This is your homework. You will see this diagram clearly and you will read all the points which are written here. I have explained it to you. Blood test is barrier. Sertoli cells, they are forming a kind of barrier between blood and the spermatogonia, because if blood has touched the germ cells, it will destroy all the germ cells by immune reaction. So it's written here, blood test is barrier. Tight junctions join the neighboring sertoli cells. Adjacent sertoli cells are joined by very tight junctions. Okay. Substances must first pass through the sertoli cells before they can reach the developing sperm. So in simple words, we can say, suppose this is blood, then there is a barrier. The barrier is formed by the sertoli cells, and then there are the germ cells, the germinal cells. So all the requirements of the germ cells are fulfilled through the sertoli cells. Whatever metabolic wastes are produced by the sertoli cells, that are back passed on to the blood through sertoli cells only. So this much you have to remember, that sertoli cells are forming blood testes barrier. Arima Pandey says, sir, your sound has tremendous amount of energy which can create new world. That is what I want, but I need your help for that, right? Today you are learning from me. So now it's your duty to tell your friends also that wonderful sessions are being conducted at Unacademy NEET Elixir, so that they can also follow our channel. Okay guys? Chali, next. This barrier prevents an immune response against the spermatogenic cell surface antigens, which are recognized as foreign by the immune system. Blood has never seen the germ cell. So if blood comes in contact with germ cells, it will destroy the germ cells. So sertoli cells ensure this thing, that blood should not cause any harm to the developing germ cells. What you can see here is spermatogenesis, right? Can you see these actively swimming cells, the spermatozoa? In a single ejaculation, millions of sperms are released into the female reproductive tract. Okay, so this is what we have to understand. Spermatogenesis is process of formation of male gametes. Understand, what is spermatogenesis? Process of formation of male gametes. What are male gametes? Sperms. When does spermatogenesis begins? It begins during puberty. Puberty is 12 to 14 years of age in case of males, and continues till death. Very important. A male continues producing sperms throughout his life Agreed. With the advancing age, जैसे जैसे उम्र बढ़ती है, the number of sperms produced as well as the quality of the sperms reduces, right? As the age advances, the number of sperms produced, the number of healthy sperms produced, that reduces. But a male continues producing sperms till his death. So here you can see the complete process of spermatogenesis, enlarged view. This is spermatogonium diploid. It undergoes growth. Shall I write here? It undergoes growth and it is forming what? It is forming primary spermatocyte. Primary spermatocyte is again diploid, 2n. It undergoes meiosis 1 and forms 2 haploid secondary spermatocytes. Chromosome number has reduced. Then secondary spermatocytes undergo meiosis 2 and they are forming these cells which are called what? Spermatids. Each spermatid differentiates into one sperm. From one spermatid, one sperm. That is called spermiogenesis. So this process, when spermatids are converted into sperms, this process is called spermiogenesis. Mature sperms are formed. Spermatid structure and sperm structure is a little different, right? We'll discuss what all changes happen when spermatids are converted into sperms. So if you are liking the slides, please hit the like button also. The entire process, spermatogenesis, can be divided into two substages. One is spermatocytogenesis and the other is spermiogenesis. Spermiogenesis, there is another term, spermatilosis. Overall, spermatogenesis can be divided into two substages, spermatocytogenesis and spermiogenesis. Now see, what is spermatocytogenesis? Formation of spermatids from spermatogonia. Spermatogonia, they get converted into spermatids. That is spermatocytogenesis, when cytological changes are happening. Then when the spermatids are converted into sperms, that is the next stage, that is spermiogenesis. Please do not get confused between these terms. Spermatocytogenesis, spermiogenesis, spermiation. So here you can see spermiogenesis is differentiation or the metamorphosis of the spermatids into sperms. We also use this term, conversion of spermatids into sperms. Clear hai ji? Ananya, Divya, Vashu, Tirthraj, great, love you all. So you can see this is spermatid, how the Golgi complex will be forming the acrosome. The acrosome of the sperm develops from Golgi complex. I will discuss the structure of sperm in detail. Acrosome develops from Golgi complex and it can be compared to a modified lysosome, enzymatic knife. It has several hydrolytic enzymes which are used at the time of fertilization. Right guys? Okay? Spermatocytogenesis. What is that? Spermatogonia multiply by mitotic divisions. You know, spermatogonium is diploid, and when it is diploid, that means 2n, right? And that is 46. This diagram is from NCERT. Important. They can ask you about the labeling. So please try to practice this diagram. I'll share the PDF with you people, but you have to practice on your own. Okay guys? Some of the spermatogonia called primary spermatocytes periodically undergo meiosis. Some spermatogonia, they have undergone growth and they formed primary spermatocytes. Primary spermatocytes mein what happens is meiosis, right? A primary spermatocyte completes the first meiotic division leading to formation of two equal haploid cells which are called secondary spermatocyte. Now when meiosis 1 has happened, the chromosome number has reduced. So from 46, the chromosome number has become 23. Okay. Secondary spermatocytes, they undergo second meiotic division, as I told you. And second meiotic division or meiosis 2 is similar to mitosis. So now the chromosome number is not going to reduce and they're going to form what? Haploid spermatids. Okay guys. Then spermiogenesis, again important. It is differentiation of spermatids. When the spermatids will be converted into spermatozoa, that is what is called as the spermiogenesis. Now this sentence is very important. Question was framed in NEET 2022 from this sentence. after spermiogenesis, sperm head becomes embedded in the sertoli cell. Sertoli cell में वो धंस जाता है, and they are finally released from the seminiferous tubule. Now students, they got confused from the seminiferous tubule or into the seminiferous tubule by the process called spermiation. So there was a little controversy also in NEET 2022 exam when this question was there, but this statement given is right. Technically it should be that they are released from the membrane of sertoli cell into the lumen of the tubule. So this diagram also is important, how spermatid is getting converted into sperm, how the flagella is being formed, how the acrosome is being formed. So this is important. Now this is a highly enlarged view of one sertoli cell. Can you see how one sertoli cell is having all these structures? I have labeled it here. What? It is sertoli cell. This is sertoli cell, this big cell with this convoluted kind of membrane, and you can see this is spermatogonia, primary spermatocyte, next stage secondary spermatocyte, next spermatid, and then these are the spermatozoa. These structures, their head are embedded into the sertoli cell. Is it clear? Beautiful diagram. Ananya says your diagrams are so clear. Wonderful, G. Hit the like button if you're liking the diagrams. All these diagrams will be made available to you people. This PDF is completely free of cost for you people. And this is the endocrine regulation, hormonal regulation of spermatogenesis, which we'll be discussing in the next class. So let me tell all of you, install the Unacademy app in your phone. Install the Unacademy app in your phone, and these days we are running special discounts also for you people. This is the daily practice paper which you have to solve on your own, and I'll be discussing in the next class the answers to these questions. Do not forget to join us on the Telegram NEET Elixir and install the Unacademy app, and you can use my code, that is Sachin Live. Soon I am going to start your batches on our Plus platform. Right guys, this is the code which you have to use, and I thank you all for this session. God bless you all. Namaskar. Bye bye. पढ़ते रहिए, खुश रहिए. मिलेंगे हम फिर से, and do not forget to subscribe to our channel and hit the like button. Wish you all the best. God bless you. Bye bye.